All right, in this video, we're going to be talking about using antibodies as medicine. And this is one of the most exciting new advances in medical technology and pharmaceuticals. Antibodies have the ability to be highly specific in the way that other drugs, like small molecule drugs, have not been able to be. And so I'm going to give a few examples and talk about the general principle of how this works. So the first thing to say is the goal of all of this is we want this antibody that we're going to inject as a medication to target something that the patient's immune system should be destroying but isn't recognizing for some reason. So the majority of cases that you see, this is used against cancer or neurodegenerative disease structures like plaques and Alzheimer's disease. And in both of those cases, the cancer or the Alzheimer's plaque, they're structures that were produced by the patient, right? They're not foreign like a bacteria or a virus. And so it's sometimes difficult for the immune system to recognize these things because they appear like part of the person. So if we want to do this, we need to to overcome a couple problems. The first one, and by far the most difficult, is for cancer anyway, we need to find a antigen, right, a protein or a molecule on the surface of the cancer cell that is highly specific to that cancer. And by highly specific, I mean it can't be found on non-cancer cells, right? Otherwise, you're going to be injecting an antibody into your patient that's going to trigger the attack of many, many cells, healthy cells inside the patient, and we can't have that happening. So this is going to require a search, right? You're going to have to take biopsies of tumors and try to find molecules, or at least parts of molecules, that are very, very specific to that cancer cell. The second thing you're going to have to do is produce an immune response so that you can actually make the antibody. Now, the patient can't do it, obviously, or else they already would have, and we wouldn't be having to deal with this cancer because the immune system would have taken care of it already, right? So the easy solution to this is we're going to use an animal to make the antibody for us. So we're going to take the human antigen that we found up here. We're going to inject that antigen into our animal, usually a mouse or a rabbit. And since that protein is definitely foreign to the mouse or rabbit because they have a different genetic makeup than a human does, they'll definitely produce antibodies. Right? Now the problem is, remember, in the structure of an antibody, you have variable regions up here where the antigen binds, but then you also have constant regions, right, where your immune system is supposed to recognize the antibody. You can't have that constant region having the DNA sequence, the genetic sequence coding for it from the animal, because then when you inject it back into a human patient, it's going to look foreign. And so the third part of this is we're going to have to somehow modify the constant region of that antibody to make it look human. We call that humanizing the antibody. And so what we're effectively doing is making this Frankenstein antibody that's half animal, half human. Right? The variable regions that bind the antigen will be from the animal, and the constant region that the immune system recognizes will be the human sequence to make sure the patient recognizes it as being, right, a normal antibody that it needs to use and recognize. All right, so for part one of this, we need to find an antibody that we're going to use in the first place. And the way that we normally do this is we immunize our mouse with proteins and glycoproteins that says glycoconjugate from the tumor, for example, if cancer is what we're targeting. Now, this mouse is going to make probably hundreds or even thousands of different antibodies that bind something, right, on that tumor sample that you injected into it. And there's no way that all of them are going to be functional. And so what we're going to do is, once that mouse has had an immune response, we're going to get cells out of its spleen. And those splenocytes are effectively the cells that would be B cells, B lymphocytes. These are antibody-producing cells. Now the problem is B cells, when you take them out of a mouse or a human, they don't do that well in cell culture. And so to make them immortalized, we take tumor cells, actually, and we merge the tumor cell with the B cell, and they make this, sort of hybrid, half cancer, half B cell that we call a hybridoma. Right? This is basically like a rogue antibody-producing cancer cell. And we don't really care that the cell's unhealthy because we're not putting the hybridoma cell inside of the patient. We're putting the antibodies, hopefully, that one of these hybridoma cells produces back into the patient. All right, so we take our hybridomas, we grow them, and we extract the antibodies off of the liquid medium that they grow in. Then you're going to use ELISA, which, if you're not familiar, we talked about that in the previous video, so you can go back and check it out. ELISA is testing our antibodies against many different antigens from the tumor. And what you're looking for is, you're looking for an antibody that sticks very well to your cancer cells in the tumor, but does not stick to healthy tissue. Right? You're going to find lots of antibodies that stick to the tumor, but that also stick to healthy tissue as well, and we cannot have those. Right? So this screening process is the most difficult part of it. We've got all these different antibodies. We want to find one that's as specific as we possibly can. And if we do find that antibody, then we have to humanize it. Right? So this is showing the amino acid sequence of constant regions from human antibodies, and this is from mouse. If you notice, they have a lot of similarity. If you try to line these up, a lot of the amino acids agree with each other, but they are definitely not perfect. And so we have to effectively take the constant region and swap out the mouse protein, right, the mouse amino acids, and replace them with the human version so that they don't get rejected when we inject them back into the patient as medication. All right, so all of this, this is the most basic, fundamental way that antibodies are used as medication. It's incredibly promising because these antibodies are much less likely to have horrible side effects than small molecule chemotherapy drugs are, because those drugs tend to be horribly nonspecific, and that's why the patient feels so sick, is because we're effectively poisoning like all of their body. We just hope it poisons the cancer a little more. Here we have the possibility of being highly specific, right, and using the immune system rather than poisonous chemicals. All right, so other variations of this. One other way to use antibodies to treat cancer is something called antibody-drug conjugates. And all that means, conjugate means to link together. And so this is where an antibody and a drug are linked together, and you're using the antibody as like almost like a heat-seeking missile finds its target. The antibody is finding the cancer, but it delivers a drug payload along with it. Right? So in this case, we have our variable regions just like we talked about. These are what would bind the antigen, and we hope the antigen is something on the cancer cell in the tumor. Now, on the constant region down here, we're going to link a drug to it, what they're calling the cytotoxic payload. And the hope is, if we design our antibody correctly, right, by linking the drug to the constant region, if the target cell engulfs the antibody by endocytosis, we've now sort of snuck our drug into that cell. But since this antibody shouldn't be binding to other random cells throughout the patient, it should give us a highly specific response. Okay? And so the hope is that our antibody gets delivered, it gets ingested by endocytosis and brought into this vesicle, and once your drug enters the lysosome, lysosomes have acidic pH, they have enzymes that start chopping up proteins. They're going to degrade and destroy the antibody, and the hope then is that the drug gets released, leaves the lysosome, enters the cytoplasm, and does whatever it does, right? So DNA intercalation means it's going to degrade DNA in the cell, microtubule disruption because microtubules form the mitotic spindle, and cancer cells like to divide a lot. And so if this is successful, we basically dramatically increase our concentration of drug in the cancer cell while keeping a very low concentration of the drug in other parts of the body because the antigen wasn't present in those other cells, and so the antibody didn't bind. All right. Okay, and lastly, something very similar to this, instead of an antibody-drug conjugate, this is showing radioimmunotherapy, which is a similar concept. It's just instead of a drug, we're attaching some sort of radioactive substance or radioisotope to our antibody. So radiation has long been used as a way to try to kill tumor cells, but you have to shine the radiation at the tumor, and it risks the radiation hitting other surrounding tissues and causing damage and mutations. And so if you can attach your radioactive payload to the antibody, you get the same effect as a drug. It's just this isn't really truly a drug. It's a radioactive emitting molecule that we hope will enter a cancer cell, damage its DNA, and kill the cancer cell so the immune system can come and clean up the damage. All right. Okay, hope you found this interesting, and follow along. There'll be many more videos to come. So flag the playlist, subscribe to the channel, and get all the updates.